Begin by labeling your tubes. The first well will be for the positive control, the second well will be for the non-template control, and the third well will be for the negative extraction control. Add 40 microliters of nuclease-free water to the negative extraction control, and then continue adding 40 microliters of each sample to the respective well. Add 3 microliters of EERTC to the negative extraction control and to each sample. Here's the D2P reagent, and you can see that there's sedimentation visible. So we're going to vortex that, and then we're going to add 10 microliters to the negative extraction control and mix by pipetting. And you're going to want to vortex between adding the D2P reagent in between each sample because it settles very quickly. Next, you're going to want to cap the samples before spinning them down. These are what the PCR reaction mixes look like. They're numbered on the top and the side. Take out a 384 well PCR plate and label it with the assay type and the date. It's also helpful to label where you're going to be adding master mix with a small Sharpie. If you have a repeat pipetter, set it to 7.5 microliters per well for the number of samples that you need. Here we are adding PCR reaction mix 1 down column 1 of the 384 well plate, and you can see that we're skipping wells in between, and that's because the spacing in 8-strip tubes and 96 well plates is double that of 384 well plates. And then you're going to go for reaction mix 2 along column 2, and you're going to fill the plate with the number of master mixes that you have for that assay. After incubation, you can see the D2P resin at the bottom of the wells. When you are pipetting your samples into the 384 well plate, you're going to want to avoid that resin at the bottom. We also call it sponges sometimes, and it can interfere with the qPCR reaction. And then we are going to add positive control, or PC, to the first well. And then we are going to add nuclease-free water to the second well. And this just makes it easier than single pipetting. So we're going to add it to the 8-strip tube so we can then go in with our multi-channel when we want to add all of our samples to the 384 well plate. Set your multi-channel pipette to 2.5 microliters. That's what we're going to be adding for samples into the 3D 4-well plate. Check your 8-strip tube so that there's no bubbles. And then it's helpful to get comfortable and set up so that you're going from the tip box into the 8-strip tubes that have our samples as well as the controls that we added directly into the 3D 4-well plate. And then you'll change tips every time since the master mixes are different. That's important to know. And you're going to repeat this process, changing tips in between each time until all of the wells are filled that have master mix. Now it's time to seal our plate. In the comments, tell me what you call this thing, this plate sealer. I've heard some people call it a squeegee. I think that's so fun. Also, just a reminder that you can never seal a plate too much. Thanks for watching. Be sure to check out alliantlabconsulting.com, and our socials are linked below. Don't hesitate to reach out with any questions that you might have. Bye!